Dr. D, 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 Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are covering Chapter 15 from our Genetics Essentials, Concepts and Connections 4th edition's book. This chapter covers genomics and proteomics, so let's go ahead and get started. When you're looking at genomics, you're thinking about the whole genome of organisms. Can you map out all of the chromosomes? Can you figure out the DNA sequence on all the chromosomes? What genes a chromosome possesses? And how many chromosomes are there? Can you map out all the different chromosomes? How are, you could do comparative genomics, where you're looking at one organism and comparing its genome to another organism, and so on and so forth. So let's go ahead and get started with structural genomics. This is the study of organization and sequence of genetic information contained within a genome. Right? Remember when we talked about Thomas Hunt Morgan? He was trying to create these genetic maps. These are the traditional way of mapping out chromosomes and figuring out where genes are with the chromosomes. These are also known as linkage maps. Remember, these approximate the locations of genes based on their relative location of other genes, based on recombination. Remember, he would take flies and then look at rates of crossing over of linked genes, and then by looking at rates of crossing over, he was able to determine how close those genes are on the chromosome, etc., etc. And then he would look at double crossovers, and he would determine that a gene is between two alleles, and by doing so, you could map out. It's a crude way of mapping out a chromosome based on the units of centiMorgans or map units, which again is distances expressed by recombination frequency. So this is not a very accurate way of describing a chromosome. Nowadays, more physical maps are made. Physical maps are actually based on direct analysis of the DNA. We're talking like doing restriction mapping and then doing sequencing. And this way you get a real good idea of the structure of that chromosome. It's not in frequency of crossover. We're dealing with base pairs, kilobase pairs, megabase pairs. It's much higher resolution than genetic maps, and this is the preferred way of studying structural genomics today. So you can see here that when you prepare DNA for these physical maps, you gotta cleave the chromosome into small segments, and then you're going to clone those small segments into individual plasmids, and then you can work with those individual plasmids in order to map out chromosomes and sequence the chromosomes. So speaking of sequencing the chromosomes, sequencing entire genomes is now a common day practice. It all started with the Human Genome Project. Sorry, that was Gizmo over there. We've only just recently, as human beings, developed the tools necessary to map out entire genomes. And I would say in the last 20 years or so, we've gone from just having a few genomes mapped out of maybe some small microorganisms to now we have all of the genetic model organisms genomes mapped out, and we're working on more and more each day. And now we can map a genome in just a few weeks. It's not even a challenge anymore. We've gotten to the point where we can map out genomes very quickly. So mapping out an entire genome presented challenges early on because of the size of a genome, especially eukaryotes. We're talking about millions upon millions or billions of base pairs. Remember that Humans have over 3 billion base pairs to map out. Sequencing cannot start at one end of the chromosome and end at the other end of the chromosome. So you can't just take a chromosome and sequence from one end to the other. It doesn't work that way. Only about 600 nucleotides can be sequenced at a time. So if this is true, you have to take your chromosome, you have to take your DNA, you have to break it into thousands or millions of little overlapping fragments and then clone those and then sequence those. See, so that's what I'm talking about here. You take your chromosomes, you cut it into millions of little fragments, and then you have to study each one of those little fragments. And then these sequences need to be sorted back together like a puzzle. And this was mastered by the Human Genome Project in the early 2000s, late 1990s, around the year 2000. The Human Genome Project was undergoing and or underway. And there were two approaches that were employed. A map-based sequencing approach, which proved to be more tedious, and a whole genome shotgun sequencing approach, which is actually the gold standard now. It's the way that was proven to be more effective and an easier way of mapping out an entire genome. So again, Human Genome Project began in October 1990 with a map-based sequencing approach. And I'll explain to you the difference between a map-based sequencing approach versus a whole genome shotgun approach. But the first attempt was a map-based sequencing approach utilized by the International Human Genome Sequencing Consortium. So here you would take two or more overlapping DNA fragments that form a contiguous stretch of DNA or a contig, and you would map based on these contigs. Let me show you, an example here. Here's an example of map-based approach to whole genome sequencing. Part 1, you take your chromosome and you partially digest the DNA. This results in overlapping fragments that you then clone into bacteria. So you see, you digest the DNA with those restriction enzymes we discussed in the last chapter. These large insert clones are analyzed for markers or overlapping restriction sites. So you analyze these clones, which allows the large insert clones to be assembled into a contig. I just told you what a contig is, a continuous stretch of DNA. So you need to know what a contig is. Again, a set of two or more overlapping DNA fragments that form a contiguous stretch of DNA is called a contig, a set of two or more overlapping DNA fragments. So again, you digest your DNA, you clone it, then you look for these contigs. You assemble these fragments into contigs, a continuous stretch of DNA. Next, a subset of overlapping clones that efficiently cover the entire chromosome are selected and fractured. Then these pieces are then cloned, right? So you clone these. Each of these small insert clones is sequenced, and overlap in sequences is used to assemble them in the correct order. 6, the final sequence is assembled by putting together the sequences of the large clones and filling in any gaps. So you see why this might be tedious, the map-based approach. You had to start with a chromosome, partially digest it, clone these parts, look for contigs, figure out how the contigs fit together, then digest it again into small pieces, into small clones, and then sequence those small clones. That's a lot of work. You don't need to know exactly how this process works, obviously, but what I do expect you to know is that this was a lot of work to digest the DNA, find the contigs, insert the clones, and digest them again into small fragments and sequence. It just sounds like a lot of work, doesn't it? Because it was a lot of work. So they even though these guys started earlier, the map-based sequencing by the International Human Genome Sequencing Consortium, they started earlier, but they were actually taking a lot of time, right? Because this is a tedious process. Okay, so while they were working on mapping out the human genome, a whole genome shotgun sequencing approach was started by Craig Venter in 1998. He was the head of a company, Celera Genomics. And what he did, he led a private effort to sequence the human genome, right? And he used a different technique called this whole genome shotgun sequence technique. It based on sequence overlap. So what he did that was different was, instead of making these contigs, which takes a lot of time and effort, he would take small insert clones prepared directly from genomic DNA, and then he would use powerful computer programs or powerful computer algorithms to assemble those small fragments back together into an overlapping sequence. So you see how that would save a ton of time. So here you can see Venter's approach was the whole genome shotgun sequencing approach, where you take your genomic DNA, your chromosome. Genomic DNA is cut into numerous small overlapping fragments and cloned into bacteria. No contigs are necessary. Each fragment is sequenced, so you just skip right to the sequence step. Overlap in sequence is used to order the clones, and the entire genomic sequence is assembled by powerful computer programs. So you see how that's a lot less work. It's almost like half the steps of the map-based approach. So because of that, by summer of 2000, both groups announced completion of the human genome rough draft and declared complete in 2003. So even though Venter, shown here on the left, started the process about 8-9 years later than the consortium, he quickly caught up and they finished about at the same time. And now, believe it or not, it is possible to sequence an entire human genome in just a few hours with next-gen techniques. So what took years before now takes hours. You know, when you send your DNA off to 23andMe, or now it's almost a novelty type thing. We're talking a few hours. So again, what was a contig? A contig was a set of overlapping fragments that form a continuous stretch of DNA. Okay, so now that we know the sequence of the genome, we can then study the sequence of the genome. What makes me different than you? What makes one human different from another? Or what makes this species of organism different from that species? Or what makes prokaryotes different from eukaryotes? Now we have all the sequencing information, so we could take a look at differences and similarities. So what did we learn? Well, one area of interest are these single-nucleotide polymorphisms, also called SNPs. SNPs, that means SNPs are sites in the genome where individual members of a species differ in a single base pair. So if I have an allele and it has one base pair in the allele, where there's a G instead of a C, that would be a SNP, right? It's a single base pair difference between my allele and your allele, for instance, in a population. Humans are 99.9% genetically identical. Humans are 99.9% genetically identical. However, we do differ in our SNPs, okay? And so we can study these SNPs, and by those SNPs, we can determine relatedness. We can determine all kinds of different, it has different implications for health, for, again, relatedness, for heritability. And we could even use SNPs to identify people. We can use SNPs to identify the causes of disease, so genes that are linked to disease. So SNPs are a big area of study today. And haplotype refers to the specific set of SNPs that someone has, so that are on a chromosome. So on a particular chromosome, you'll find certain areas of SNPs, these single nucleotide polymorphisms. And the host of SNPs for a chromosome, okay, are called the haplotype. Okay, SNPs within a haplotype are physically linked, obviously, because they're on the same chromosome, so they tend to be inherited together. So when you do these genome-wide association studies, you can use SNPs, okay, and you could figure out which SNPs are responsible for diseases, for instance. Like they did a study with, I believe it was high cholesterol, and what they did was they would take people with high cholesterol, people without, and they would study their genomes for SNPs, and they would determine, okay, this particular polymorphism is found in a disproportionate amount of people with high cholesterol versus low cholesterol. You see what I'm saying? So that would direct the researchers to particular genes that they could further study to see if there's a link between those genes and their particular disorder, like high cholesterol. Isn't that interesting? So we may not know exactly what's causing high cholesterol, but then by studying SNPs, we could go look for maybe alleles of genes that are mutated by small polymorphisms, and then we could get insights into which genes might be responsible for their hypercholesterolemia. So here's an example of what I'm talking about. You can have the chromosomes are identical for most DNA sequences. So you have chromosome 1 from different people. You have chromosome 1 from different people. I noticed that the sequence is the same except for these SNP sites. So here's a SNP site, so 3 different SNP sites. Each haplotype is made up of a particular set of alleles at each SNP. Okay, so these are all from different people in a population. So if you were to find, for example, that, let's see, this person, let's say person 1B had high cholesterol, you might say, well, if A, C, and D, these people did not, well, here's a candidate right here. This SNP here, you see how there's an A instead of a G in chromosome 1B, and in this person, I might say, well, this actually might be responsible for their cholesterol, right? So that's a candidate that you could further study, and that may or may not lend to their genetic disorder, right? So that's kind of interesting. Another area in genomics is called bioinformatics. Scientists who study bioinformatics kind of bring together the two worlds of biology and computer science. They use molecular biology and computer science in order to compare genomes, as comparative genomics, in order to do the types of SNP studies I just told you about, studying SNPs and trying to link those SNPs to various diseases. They look at evolutionary markers. They study how different speciation events may have occurred. They could study, I told you about microarrays, microRNAs. They could study the genome to find candidate sites for microRNAs that we may not have discovered before by looking at, consensus sequences of microRNAs. So many things you can do with a strong computer and biology, because now that we've sequenced the entire human genome, you can actually go through and make algorithms, computer algorithms, to try to parse out areas of interest, right, that may not have been discovered in the genome. Metagenomics, here's another area. Sequencing genomes of entire communities of organisms. Here's another new area of research. So for example, one hot area is microbial communities on the human microbiome, like your gut microbiome, for instance, or your skin microbiome, looking at all the different organisms and, comparing the genomes of them on the microbiome of your gut, trying to figure out, okay, if someone has gut issues, is their microbiome different than someone without gut issues? Or if someone has skin issues, is the skin microorganisms biome different? Those genomics, are they different? looking at neat questions like that. Synthetic biology is really interesting. Creation from scratch of novel organisms. They're trying to create synthetic chromosomes. They created a whole synthetic chromosome 3 for yeast, for instance. They're trying to make synthetic chromosomes for bacteria. They're trying to create life in a test tube, which is really neat. What else? Functional genomics. What about people who study functional genomics? Characterize what sequences do, right? So what do different sequences do in the genome? Okay. The transcriptome. People studied the transcriptome. That's what are all the RNA molecules in the genome? The transcriptome means all of the mRNA in a cell, right? All of the mRNA in a cell, which should be reflective of all the gene expression that's going on in a cell. And as you remember from a previous chapter, not all genes are expressed all the time, right? A certain subset of genes are expressed. So those genes will have mRNA in the cell. And so the transcriptome would be all of those transcripts, all of the mRNA for those genes in the cell. The proteome is all of the proteins in the genome, in the cell. So the proteome, what proteins are available in the cell? Here's another interesting area of study, gene expression with microarrays. Microarrays are fascinating. What microarrays let you do is figure out gene expression profiles of cells. Gene expression. What does gene expression mean? Remember, a gene turns on, you got transcription, right? Translation, transcription. Specifically, microarrays look at transcription. If a gene turns on in a cell, you're going to get more transcription, right, in that cell. If a gene turns off in a cell, you're going to get less transcription in a cell. So microarrays are a technique for looking at gene expression changes over time in a cell, all right? Gene expression differences between cells, right? So look at this. Here's a microarray. What you have here are chips. This is called a microarray chip. And this is what the results are going to look like. You're either going to get green areas or red or yellow. Okay, green, red, or yellow. Now, what you have on these chips are pixels. Let me try to zoom in. You see those individual pixels there? You got individual pixels on this chip. Each one of those pixels represents a different gene, right? So here's a gene, here's a gene, here's a gene. So each row, so look, each spot represents the expression of one gene. So each spot, each pixel is a different gene. So you'd have thousands of different genes on here, right? And let's see here. Each row is from a different tumor patient. So, okay, let me explain it this way. Each one of these pixels is a gene, right? It represents a gene. So you've got different pixels with genes. You've got DNA probes, gene probes on each pixel. Here's each pixel. A microarray consists of DNA probes, these gene probes, fixed to a solid support such as nylon membrane or glass slide. Okay. Number 2, each spot consists of a different DNA probe, which means a different gene probe, a different gene. Now, let's say you want to figure out the difference between a cancer cell and a normal cell, right? So you want to know this cancer cell, what genes are maybe upregulated in the cancer cell? What genes are downregulated in the cancer cell? This array will answer that question. Isn't that fascinating? So what you'll do, you'll take cancer cells, break them open, get their mRNA out, reverse transcribe, remember the enzyme reverse transcriptase, reverse transcribe the RNA to DNA, remember that's called cDNA, and this cDNA has a red fluorophore, red fluorescence. You do the same thing with normal cells, right? Non-cancer cells, healthy cells. You crack them open, get the mRNA out, reverse transcribe that to cDNA, but this time with a green fluorophore, green fluorescence. Okay? Then you hybridize. You add a mixture, you add the mixture of these cDNA with the red fluorescence and the green fluorescence. You add that cDNA to the probes and it will anneal. There's hybridization. The hybridization occurs between the gene that's stuck to the slide and the cDNA that was created from the mRNA in the cell. If more of the cancer cell cDNA hybridizes, you'll get more red fluorescence. If more of the non-cancer cell cDNA hybridizes, you'll get more green fluorescence. If it's an equal amount of both, you'll get more yellow fluorescence, because yellow would be a combo of green and red. Okay, so then what you can do is look at this chip. Look at this chip. So anywhere there's green, you know what that tells me? That tells me that the non-cancer cells, the healthy cells, express more of these green genes than the cancer cell. And do you see the red spots? Those are genes that are upregulated. These red spots are genes where transcription has been upregulated for those genes in the cancer cell. So, you know, if I'm a researcher, what I'm going to do, I'm going to say, well, something's causing this cancer, and it's most likely some of these really highly upregulated genes in the cancer cells. Like, these are genes. I'm sure one of these red genes, one of these red color genes, is responsible for this cancer cell. Or maybe two of them combined is responsible for this cancer cell. Now I have what are known as candidate genes to study if I'm trying to figure out what's causing that cancer, right? I know what genes have been upregulated in those cancer cells. And maybe by knocking down those genes or making a drug, once I discover what gene is responsible for this cancer, I could make a drug against the protein of that gene product and maybe treat that cancer. Isn't that interesting? So that's kind of how it works. Isn't that fascinating stuff? So here you could do the same thing, but with microRNAs. Remember microRNAs? I could look at microRNA expression in normal cells and compare it to cancer cells, etc. So I could even look at microRNA expression differences between normal cells and cancer cells. It doesn't have to be just mRNA differences in normal cells versus cancer cells. Okay. So then we get into the area of comparative genomics, comparing prokaryotes with one another, comparing eukaryotes with one another, comparing populations with one another. Let's start with prokaryotic genomes. So prokaryotes, what do we know about them? The genome size and number of genes has a positive correlation. What does that mean? Normally, every 1,000 base pairs of chromosome, for every 1,000 base pairs of chromosome, you find about one gene in a prokaryote. Okay, so that's what we mean by the genome size and number of genes has a positive correlation. There's about a gene every 1,000 BP. Okay, and remember, there's horizontal gene transfer. There's no sexual reproduction. There's only horizontal gene transfer. Remember, genetic exchange of genetic information with that sex pilus we talked about through conjugation. Okay, and then the function of genes. Only about half of the genes identified in prokaryotic genomes has been assigned a function. So we still don't even know what half of the genes of a prokaryote do, right? Isn't that interesting? And a quarter having no significant sequence similarity to other known bacterial genes. So there's like between E. coli and Salmonella, a quarter of those genes don't even have any sequence similarity. So it makes it exceedingly difficult to do comparative genomics. So here you can see different prokaryotes, right? The prokaryote with the fewest base pairs in its genome is this guy. I'm not even going to try to pronounce this guy. Nanoarchaeum equidens. Okay, that wasn't so bad. Nanoarchaeum equidens, which only has about what is this? Oh, only like 490,000 base pairs in its entire genome. And it has about 536 total genes. That's interesting. And this is the microorganism, or I should say prokaryote, with the biggest genome. This one looks even harder to pronounce. Bradyrhizobium japonicum. Okay, this guy has 9,100,000 bases, right? So that's a pretty big genome for a bacteria. Okay, and this guy has 8,317 genes. But E. coli is more typical of it's more typical for a bacterium. It has about 4.6 million base pairs and about 4,289 genes. Okay, so again, the bigger the genome, the more genes for a prokaryote. And again, you see these are the functions that we've assigned to the genomes of prokaryotes. Notice this wedge, this part of the pie, number 2. That's unknown function. We still don't know the function of a lot of these prokaryotic genes. Concept check number 2. What is the relationship between genome size and gene number in prokaryotes? It's positively associated, right? Species with larger genomes generally have more genes than species with smaller genomes. So there's a positive correlation. So gene density is relatively constant. Now let's discuss eukaryotic genomes. What's the difference there? Genome size and number of genes have no correlation, okay? So just because you have more DNA for a eukaryote does not necessarily mean at all that it has more genes or more important information. There's no correlation there. Genome size is not correlated with organism complexity either. So just because you have more DNA doesn't make you more of a complex creature. The number of genes is not correlated with organism complexity. Okay, so the number of genes isn't even correlated with complexity. So amount of DNA is not correlated with organism complexity. Number of genes is not correlated with organism complexity. Interesting. Eukaryotic genomes contain multiple copies of many genes due to evolutionary gene duplication over time. So you might have several copies of the same gene. In more complex eukaryotes, both the number and length of the introns are greater. So one thing we do know is in the more complex eukaryotes, the number of introns you'll find and their lengths seem to be higher. So for humans have the most numerous and longest of introns of other organisms. Isn't that interesting? So here you can see what I'm talking about. Here's genome size, and here's the predicted number of genes, and you're not going to find much correlation. So here there's only like 12 million base pairs in Saccharomyces cerevisiae, the baker's yeast, only 12 million base pairs of DNA, only 12 million base pairs of DNA, and you got 6,000 genes. But compare that to, let's compare that to Zea mays, corn. We've got 2.4 billion nucleotides, and you have 32,000 genes. So you see how there's really no correlation between amount of DNA and number of genes, or amount of DNA and complexity of a creature. Because look, Homo sapiens, humans have 3 billion base pairs, and that's, corn has 2 billion, and corn has way more genes than we do. They have 32,000 genes for corn, and we have only about 25,000 genes in humans. And yet, we are more complicated creatures. We are more complex creatures than corn, for instance. All right, so what else do we know from eukaryotic genomes? Non-coding DNA is a big deal, right? Non-coding DNA takes up 98.5% of the human genome. The rest of it I mean, the non-coding stuff is called junk DNA. A lot of scientists still refer to it as junk DNA. Genetically engineered mice missing much of this non-coding DNA appear to have no abnormalities. So, there's two areas of thought right now. Some scientists claim that junk DNA is just junk, right? It's just, because if you engineer it out of mice, they still live perfectly normal, happy lives, right? And then other scientists will say, well, it's not junk because it could be the reservoir for enhancers. That it could serve as enhancers, or it could serve as some kind of chromatin remodeling areas, or areas of heterochromatin, areas of chromosomal stability, etc. So make of that what you will, but there's strong evidence for both. There's evidence that a lot of it really is dispensable. It's not really important, as evidenced by actual studies like genetically engineering mice, where you've chopped out millions and millions of non-coding base pairs and the mice grow up just fine. And then there is something to be said about the argument where this junk DNA isn't junk, it serves as enhancers or promoters, or some other kind of interesting thing. 98.5% of your DNA does not code for a protein. Isn't that interesting? What else do we know about eukaryotic genomes? Transposable elements, which we haven't touched on too much in this course, but transposable elements are DNA sequences that can move about in the genome. They can kind of pop in and pop out of parts of chromosomes. These are responsible for a substantial part of the genomes in multicellular organisms. So 45% of the human genome is derived from transposable elements, although now they're mostly defective. They've mutated to the point where they can't transpose themselves. They can't hop out of chromosomes and pop into other chromosomes. And they consist of moderately and highly repetitive sequences. So you can see here the percentage of a genome consisting of interspersed repeats derived from these transposable elements. So in Arabidopsis thaliana, about 10% of the genome was formed by these transposable repeats. Zia Mays, 85%. Homo sapiens, almost 45%. Isn't that interesting? So much of our DNA is just these repeats that jumped around during evolution. Okay, a couple more points about the human genome specifically. Fun facts: human genome, 3.2 billion base pairs. Only 1.5% encodes for protein. Only 1.5% encodes for protein. Average gene length is about 27,000 base pairs with about 9 exons. So your average gene in your body is about 27,000 base pair long with about 9,000 exons. Though one exceptional gene has 234 exons. So you can actually have a gene with 234 exons. That's really fascinating. Human introns tend to be much longer and more numerous than other genomes. Remember, the more complex the organism, the longer and more numerous its introns tend to be. The function of many genes is still unknown, area of study. And a single gene often encodes for multiple protein through alternative splicing. Remember, we discussed alternative splicing before. This is when the exons are rearranged such that you get different protein products from the same gene. Each gene encodes on average 2 or 3 different mRNAs. Isn't that interesting? In your body, each gene codes for, on average, 2 or 3 different mRNAs based on this alternative splicing. Here you can see what we were talking about, humans shown in yellow. This is the length of our introns, the length of our introns. So you can see that our introns tend to be longer than other organisms like the roundworm, the nematode, or the fruit fly. Fruit flies and nematodes have a lot of very short introns. They outnumber us in short introns, but we outnumber them with long introns. And just like before, look at this big wedge here, number 17. Look at this, 17. Molecular function unknown. We still don't know the function of so many human genes. There's still so much to learn about genetics. Okay. Now I think we forgot to take our break time with Gizmo and Wicket. So let's go ahead and do that real quick, and we'll come back strong to finish this chapter. What do you say? Hey everyone, welcome back from break time with Gizmo and Wicket. We have just a few slides left in this chapter. So again, we're talking about studying the entire genome, but you could also study the entire proteome. The proteome is a complete set of all proteins found in a given cell. And the study of the proteome is called proteomics. And a common tool that's used in the study of proteomics is the mass spec, or mass spectrometry. This determines the molecular mass of a molecule. So here's how a mass spec works. You have a protein. These dots represent the amino acids of a particular protein polypeptide chain. And you treat the protein with an enzyme trypsin, and it cleaves the protein. I believe it cleaves at lysine residues, which breaks it into short peptides. You then put the peptides into an accelerator, and then from that, the readout is the peptides are analyzed with a mass spectrometer, which determines their mass to charge ratio. So you put them in an accelerator, the mass spec determines its mass to charge ratio, and that detector reads out counts from mass to charge ratio. Peaks are created. A computer program compares those peaks, compares the profile with those of known and predicted proteins, and a match identifies the protein. So you've identified the protein. Nowadays, you could get fancier with mass spec as well, so you can identify the exact protein residues. It gets very fancy, very cool stuff. But again, proteomics, looking at the complete complement of proteins inside of a cell. There are tools that study that. So nowadays, things have progressed so much in just 20 years. We've completed the human genome. We're looking at the transcriptome, which is, again, the transcriptome is all of the RNA transcripts, the mRNA or microRNA transcripts in the cell. Looking at the proteome, right? And we're studying every facet of the genome, from the genes to the transcripts to the proteins, all along that gene expression pathway. Okay, and lastly, protein microarrays are coming about. This is similar to the gene microarray I told you about, the transcription microarray we talked about, but it's with proteins. So you either stick proteins on a glass slide or filter, or antibodies for the proteins are applied. And then when there's binding that occurs to a protein, there's fluorescence. Fluorescence occurs, and that indicates the presence of a protein in a tissue. So just like we have microarrays for mRNA, now we have microarrays for proteins, another tool to study proteomics. Very cool stuff. So again, an interesting chapter about what all's been developed and discovered in the field of genomics. There's a lot of exciting stuff going on, and like I showed you before, there are still so many unknown genes in the human genome and prokaryotic genomes that it's still ripe for the picking. There's still so much to learn, and there's still so much to discover. I think there's going to be some more interesting discoveries to come. Obviously, some of these genes are going to have some really cool implications. Anyway, I hope you enjoyed the chapter. If you have any questions, throw them in the comment box below, and I'll catch you guys next time.
Dr. D, 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 Dr. D.